It is my great pleasure to introduce today's Wednesday afternoon lecturer, Dr. Yishan Zheng. Dr. Zheng is the chair of the Department of Embryology at Carnegie Institution for Science in Baltimore, and she's interim co-president of the Carnegie Institution. She's also an adjunct professor of biology at Johns Hopkins. Dr. Zheng earned her Bachelor of Science degree in biology from Sichuan University, where both of her parents taught. She's the daughter of two academics, but grew up during the Cultural Revolution in China, and so had some interesting experiences. And I was actually just listening to a video that she made about how she became a scientist, and it is fascinating. After her undergraduate education, she moved to the United States to study with Berl Oakley at the Ohio State University, where she got her PhD in molecular genetics. And then she did a postdoc at UCSF with Tim Mitchison and Bruce Alberts, where I suspect she got interested in microtubules. She was recruited to the Carnegie Institution in 1996, where she works on a variety of issues related to the cellular cytoskeleton and mitosis, nuclear function, on issues related to microtubules, lamin, aging, morphogenesis, cell division, and disease. Probably partly because of her upbringing in China, where she grew up wanting to be a great writer. She has produced a series of really elegant scientific papers describing her journey through the microtubule organizing centers, including her initial discovery that gamma-tubulin was well conserved in all eukaryotes. And she discovered the gamma-tubulin ring complex as an important initiator and organizer of microtubules, no matter what their function in eukaryotic cells. She's gone on to make many other discoveries as well. Dr. Zheng was a Pew Scholar, a Howard Hughes Medical Institute investigator for over a decade, and the winner of the ASCB Women in Science Award. She has served the scientific community in many different ways and been recognized for her service as a council member for the American Society of Cell Biology, chair of the International Affairs Committee for the American Society of Cell Biology. She's a national associate of the NRC Council for the National Academy of Sciences, which is an honorary lifetime appointment for extraordinary service. And she's a senior scholar on aging for the Ellison Medical Foundation. Today, she will talk about her research on cell division and genome organization in development and aging. Please join me in welcoming Dr. Zheng.
Very much. Thanks, Michael, for such a nice introduction, and thanks to my host, Yamini, for arranging this amazing opportunity to come here, talk to people from the broader scientific community at NIH. So I'm just going to try to dive in. Where's my, oh, here. So what I'm going to do is I'm going to try to give highlights on the work that has been published. And then I'll spend the last 10 or so minutes mostly focusing on some of our most recent work on genome organization. Okay, so here it basically shows you a picture of our building. Our department is pretty small. We have 8 faculty plus 2 staff associates, but it's a pretty amazing department to be in. So I'm a cell biologist. I start my career trying to understand how cytoskeleton work, mostly focusing on microtubules. These are interphase cells, and it really depicts the complexity of an interphase cell with so many connections of a nucleus, and this is a nuclear lamina that connects chromosomes through the inner and outer nuclear envelope proteins to various forms of cytoskeleton. And then, of course, membranes are also very highly connected to one another. So the thing for me when I start my career is really trying to understand, sorry, I need to go back a little bit, trying to understand how, you really handle when you want to go into cell division, how you go from one cell to two cells. So a lot of what I'm going to talk about actually stem from our interest in cytoskeleton. So what I want to do is to first give you a highlight on our study of mitotic spindle matrix in cell division, and then I'm going to spend most of my time talking about nuclear lamins by highlighting some of our studies on how nuclear lamins are involved in tissue building and in aging, and spend more focused time on how lamins could really regulate 3D genome organization, which in turn influence transcription globally. Okay, starting from the beginning of my career. So I was, as I said, I was very interested in microtubules and how microtubules is built into a bipolar spindle. The field for decades have been mostly interested, rightfully so, focused on understanding how microtubules nucleated from the centrosomes capture the kinetochores and then ensure equal chromosome segregation and make two cells. But when I started my own career, I felt like there probably is really a need to try to understand about cell division that's beyond how microtubules capture chromosomes, because cell division is really not just segregating chromosomes. You need to segregate so many other things. There's massive changes of a nucleus, cellular membrane, including nuclear membrane. The nuclear lamina disassembles, and then it reassembles. So this reorganization really was much less understood. So when I start my lab, we've decided, okay, let's just see if we could broaden mitosis research to include other structures. Is really, what if they really have interface functions? How can you separate the interface function away from mitosis function? And we are lucky because there's a system that was developed in the 70s by people like Masui and Luca. They showed that you can take Xenopus sperm, you can isolate them, put it into Xenopus eggs, and then at the tip of the sperm, there is this centriole that will recruit materials to mature into centrosomes, make asters, and then with time, chromosomes condense, make mitotic-like condensed chromosomes, and then mitotic spindle can be established. Because this is an in vitro system, you can actually make antibodies and deplete proteins from it, and then ask what happens to this process. And if you see a defect, you can try to rescue it and then see if this defect is really caused by the protein. So even if your protein has an interface function, you can say it also has mitotic function because this system never really gone into interface. So this had turned out to be very productive for us. Using this system, we have been able to establish many nuclear proteins, as a few listed here: RanGTPase, lamin-B, and RUV, which is actually a chromatin remodeler. They have very well-established interface functions, but they also regulate mitotic spindle in various ways. And so are some membrane proteins as listed here: epsin, P97, CDC48, and FAM. FAM actually is another name for a deubiquitination enzyme called USP9X. So these proteins have interface functions, and then when cells go into mitosis, they have mitotic functions. So then the question is why you have so many moon-lighting proteins in both the nucleus, and the light is kind of shining on my eyes, in both the so many nuclear proteins as well as membrane proteins regulating mitosis. I think it really comes back in a way addressing the initial question I posed, that is, when you think about cell division, if you think beyond equal chromosome segregation, what you have left is how do you segregate the rest of the cell? Is that completely random? You vesicularize, you just dissolve and have it equally distributed? Or actually beyond microtubules, you have another network that needs to be organized around the spindle and permeating throughout the spindle. That's how you can actually properly dividing them to prepare the cell for the next cell cycle and for various things in the next cell cycle the cells might want to do. And in fact, in mitosis field for years, for decades, starting in the 60s, Bob Goldman and colleagues were one of the first to propose maybe mitotic spindle is not just made of microtubules in terms of its structure. And here's a sea urchin spindle under EM. So these are fibers of microtubules. But beyond this microtubule-like fiber, there are these granular structures surrounding the spindle. And then actually in some other EMs, you can see electron-dense materials permeating throughout the spindle. So this is where the spindle matrix idea came from. But it has never been popular. People come in and out. This is not really even a field. But it seemed to really make sense that if you want to build such a complicated structure to divide the cell, you probably need things beyond the microtubules. You probably need to organize cell with another way to kind of make the cell division to happen effectively. So the kind of initial idea has been, maybe mitotic spindle, this mitotic spindle matrix could regulate spindle morphology. It could interact with microtubules to make the spindle much more effective. So now, after we realize actually this, the cellular, other cellular components such as nuclear remnants and membranes could be playing a role in mitosis. So the hypothesis is really perhaps the mitotic spindle matrix is really the way the cell uses to organize these materials to crosstalk with microtubules to ensure proper chromosome segregation and perhaps even proper cell fate determination. And indeed, we have been able to look at this, and that's something I'm going to highlight more. Okay, so to really get a handle on what are these spindle matrix materials, what kind of proteins are in them, a former postdoctoral fellow, Min Cai, had devised a very nice assay, which is this putting Aurora-A, which is a kinase, mitotic kinase, onto the magnetic beads. And if he puts this magnetic beads in Xenopus egg extract, adding RanGTP, he's able to induce aster formation very quickly in the first, in the first 3 minutes, and then by 15 minutes you get very nice bipolar spindle assembly. So so you can see aster. This is just a low resolution view. You can see the spindle assembly. It's a wider view. This is a close up. The advantage of this is then you can use this to isolate the spindle by putting the reaction on a magnet, and then it would collect the magnetic beads, and then you just wash the spindle with nocardazole that depolymerize microtubules, and what you have left would be the spindle matrix. And we just did proteome analysis in collaboration with John Yates' lab, and that led us to find about 600 proteins. And the major criticism of this work really has been, well, we found a lot of things. While we actually hypothesized it ought to be a lot of things, but biologists generally like to see things that's pure. And if we see a lot of things, a very obvious suspect is, well, you just isolated the cell, and you have not done much. Maybe you've isolated something that's aggregated. So, but we just decided to forge forward and see what we get. And one thing we spent time on is lamin-B, because we thought this is a network and probably have some structures, and lamin-B is an intermediate filament protein, so perhaps it could provide some structure to it. We also and this is actually how we studied epson, and then today I will just mostly talk about lamin-B and another protein called, it's a zinc finger protein, we call it BugZ. I will give you some highlights on our findings about these two proteins' role in spindle assembly. So for lamin-B, over the years, and these are the published work, I'll just summarize. Over the years, we've seen that the spindle matrix protein lamin-B, they are in the spindle matrix, and it seems to be interacting with dyneins to help for a number of functions. One function is centrosome separation during the beginning of spindle assembly in prophase. So if you have nominal cells, the centrosome don't separate quickly. It also seemed to be involved in spindle orientation, and one example is in the ventricular zone in the neuronal progenitor cells. If you have lamin-B mutations, the spindle you end up finding a lot of oblique orientations when actually you should find either parallel or perpendicular orientations. We also, using Xenopus egg extract, we showed if you don't have lamin-B, you have spindle morphology defects. And it seems all of these functions had to do with the interaction between lamins and dining system. That's the best explanation for these phenotypes. So the problem we have over the years has always been that, this lamin-B or the other spindle matrix protein that I will talk about soon, when you look at it, it's really not like microtubules. Microtubules look really beautiful, whereas lamin-B looks hazy. It's both in the cytosol and it's on the spindle. And then this other spindle matrix protein is also, it's enriched on the spindle, but it's also kind of hazy. And we've really not succeeded to identify any discernible filamentous structure under EM. So then it's really hard to move forward to really understand if the structural feature of this, is there a structure or is it just some random aggregates? So that has been the biggest problem we had faced. So, but we were pretty lucky in the sense of having identified this protein called BugZ. So this is a spindle matrix protein we identified as BugZ. I'll just give a little bit background that's published in this paper. We found this protein here is basically a plot of this protein. The N-terminus, this little bit, so this is a disordered disposition. So anything below 0.5 are ordered, considered ordered. Anything above that is disordered. And as you can see, this region is highly ordered. And in fact, this corresponds to predicted 2 zinc fingers. What we found is this region binds to tubulin and microtubules. And at the C-terminus, it's essentially largely disordered. But it has a very interesting motif called Glibb's motif, which is a motif that binds to proteins such as BUB3. BUB3 is a kinetochore protein that regulates chromosome alignment and chromosome segregation. So basically through this studies, we've defined that BugZ in mitosis definitely has a role in regulating chromosome alignment by binding and stabilizing BUB3. So this is basically one function in mitosis. But the real interesting thing about it is that there is this really large disordered region. And then we are able to make a large quantity of soluble proteins that offers us a means to do biochemistry. And why that could be so interesting? Well, that's because in addition to these solid polymers such as actin and microtubules, you've got other polymers I'm sure many of you have heard, the hydrogel and the liquid droplets. And these polymers have water in it. Hydrogel has water in it, and then this liquid droplets have even more water and even more dynamic structures. And these features, and now it's a vibrant field now. These are called membranous organelles that could help organize the cell. And it really makes sense to us if you think about spindle matrix, because when you go into mitosis, many membrane-bound systems actually become vesicularized, but you still need to organize the cell. Perhaps mitotic spindle matrix could be a membranous. There are features of membranous organelles, and then most of these systems actually are formed, most of these like hydrogel, liquid droplets are formed via weak multivalent interactions of proteins that either have these repeats that can interact, or they have low complexity and they are disordered proteins. And the way they actually interact is still not very well understood in its polymerization, but it's known that aromatic residues such as phenylalanine and tyrosine, or hydrophobic residues, or polar or charged residues, they can all drive the formation of this phase-separated state. So this is called phase transition or phase separation. So now I want to show you what Bugsy looks like. Bugsy really, so here's the 2 zinc fingers. And if you look at Bugsy, it essentially has everything I've just listed. It has phenylalanine, tyrosine throughout the molecule, about 13 of them. So this top is mouse, bottom is Xenopus, Bugsy. In addition to that, it has a lot of prolines. And then what's interesting is every proline seems to be really happening right next to highly hydrophobic residues throughout the molecule. So the molecule is fairly hydrophobic, yet it's very soluble. And we think probably the proline is preventing it to completely collapse. So this gives us a sense maybe this protein would form structure not like a solid polymer that we were trying to look for. Maybe it's actually forming some kind of phase-separated state that could function in the spindle matrix. Indeed, we've tested many conditions because we have charged molecules. Well, one thing I didn't show you, let me just go back quickly to show you, is that in addition, you can see this highlights. So this is proline-rich regions generally is fairly hydrophobic. Those are highlighted in green. And then you have other regions that's actually pretty polar, concentrated with polar molecules, and then this is pretty charged regions. So that's what this protein looks like. 2 regions of proline-rich and hydrophobic. 2 regions are charged or hydrophilic. So it's kind of hard to predict what kind of phase separation characteristics it would have. And through a lot of work, we realized it actually undergoes phase separation in a concentration and temperature dependent manner. It's kind of like this elastin protein. So basically, if you increase the temperature, increase protein concentration, it will form this beautiful droplets. This is a fusion protein between YFP and Bugsy. And it's highly reversible. Of course, there's a little bit not completely depolymerizing, but it's really repeatable and reversible. It's a very nice phase separation protein. And when this was found, actually, this is the first protein that you have a protein, native protein, a full length that undergoes phase separation besides elastin. Although a lot of studies on elastin is using fragments, and this protein by itself undergoes phase separation. So we did quite a bit of mutagenesis and then tried to measure the mutagenesis on phase separation, because people have already found in some other phase-separated proteins you can actually mutate the phenylalanine and tyrosine. So we mutated different amounts of phenylalanine and tyrosine, and we also got rid of the N-terminus to look at the impact on phase separation. And the take-home message really is that Bugsy phase separation mutant are defective in promoting spindle and spindle matrix assembly. So that's kind of nice, because that at least says, well, there is a structure. The spindle matrix could be in part, the structural integrity could be in part mediated by phase separation. Okay, so I'm just going to try to end this by kind of providing one way that we believe Bugsy could promote spindle assembly is that, as I said, the N-terminus of Bugsy bind to tubulin. And when you form phase-separated state, you can see a very nice concentration of tubulin. And when you get rid of the N-terminus, so you don't bind to tubulin, it doesn't concentrate tubulin. And if you use this 2 mutant to try to do microtubule-nucleation assay, you can see that this tubulin binding is very important for microtubule assembly. In addition, tubulin binding is also tubulin binding site is also microtubule binding site. And that's very important for bundling, making microtubule bundles. And you can see in this field, you have microtubule polymer and you have Bugsy. It's just like liquid flowing along the microtubules. So I think this kind of bundling probably is pretty flexible, and that probably is important for you to glide microtubules apart, because it's not a solid to make microtubule bundle in a very big solid state. Okay, so this is basically the take-home message. I'm jumping a lot because I want to go to other things I want to talk about. So basically, the take-home message is that Bugsy undergo in vitro phase separation, and this blue region is microtubule tubulin binding site, and red region is unstructured. Red region, the hydrophobic residues and also those aromatic residues are important for phase separation. The reason we think hydrophobic residue is important is when you increase the temperature, it's kind of a denaturation, so that kind of stretches the molecule. It basically prevents intermolecular interaction, promote intermolecular interaction. In fact, we've mapped the regions of the molecule that can interact with one another. So it promotes intermolecular weak bivalent interactions, and that's what's driving phase separation. Now, when you have microtubules, the microtubule really helps promoting phase separation along microtubules. You can see microtubule droplets along the Bugsy droplets along the microtubules, and because Bugsy could concentrate tubulin and could bundle microtubules, that really is one of the reasons it can promote spindle assembly. So that's one function, and more recently we found Bugsy also can concentrate Aurora-A kinase and help activating Aurora-A in mitosis. That can also contribute to spindle assembly. Okay, so that's the highlight of the mitosis study that we're doing in the lab. Now I'm going to switch gear to talk about nuclear lamins, and I'm going to first highlight the role of nuclear lamins in tissue building. So as I said, really, it's our study of mitosis realizing, oh, lamin actually interact with microtubules, got us to pay attention to lamin's role in interphase. I was not interested in interphase at all in the beginning of my career. What really caught my attention is, oh, this system is actually quite amazing. In interphase, lamins form this polymer right underneath the nuclear membrane, and this meshwork interacts with microtubules, interacts with chromosome on one hand, and then through these nuclear envelope proteins, it's connecting chromatin with cytoskeleton. And that, to me, is quite amazing. It almost like it's very similar in concept of how the cells are, you use similar concept to solve interphase and mitotic problems. And this, in this case, it probably could be organizing interphase chromatin. And in this case, it's making sure you can organize spindle to separate the chromatin. So I see that to be very interesting. That's one reason we started to want to look at lamin, because this connection is quite interesting. So here I already had it as written down as lamins are a major structural component of the nuclear lamina, and mouse there are multiple lamins, B-type, 2 B-type, 1 A-type. Drosophila has 1 B-type, 1 A-type. The problem when my lab first started to pay attention to lamin, really one problem really is lamin. Many people really believe lamin is absolutely essential for animal cells. If you don't have lamin, you probably won't have a good nucleus. You probably won't be able to have a cell. So and then our contribution of understanding that lamin also plays a role in mitotic state that kind of further strengthen lamin is really very important for cell proliferation, for basic cell survival. But if you really look at the literature, it's really not clear that lamin plays such kind of a role because there's a lot of redundancy. People did some RNAi work or genetic work. If you look at when you look at C. elegans, there's only single lamin gene, you still have maternal supply. So basically the literature tells us it's not really certain lamin is absolutely essential for cell function. And the way when we appreciated that lamin's role here really is connecting chromatin to cytoskeleton, it's kind of indicating maybe lamin is involved in kind of morphogenesis because cytoskeleton are involved in morphogenesis. So what a very talented and brave postdoctoral fellow came to my lab, he decided, okay, I'm just going to see if I can create any cell that can be laminal, and that will demonstrate whether lamin is essential or not essential. And he started with ES cells. And the reason I say he was brave is because many seminar speakers came through my department when I told them that's what we're doing. They were saying, well, what are you going to learn if you don't get any cell? you would just learn, okay, lamins are essential. But, we just forge forward. In his time, he just did very standard mouse knockout, created double knockout of B1, lamin B1, B2, derived ES cells, used 2 rounds of deletion to get rid of lamin-A. And he was able to generate lamin TKO ES cells. And that was, a very definitive answer that lamin is really not essential for basic cell function, at least not in ES cells. And the other thing is, once you have this mouse model, you can really look at the lamin in different organs. And the major take-home here is every organ we looked at, there are defects. The brain is most dramatically affected. But it's very hard to try to get at the mechanism using mouse. So another postdoctoral fellow came to my lab now probably 5-6 years ago. He said, well, I'm going to use Drosophila as a system because it's much faster. He focused on testes, so this is the phenotype. Basically, there's one lamin, B-type lamin in Drosophila, and when you don't have that B-type lamin, the testes is very small compared to the wild type. And I will just quickly highlight what he's done. So what Haiyang has done. So basically, this is the tip of the testes, and at the tip of testes, you have 3 types of cells: the hub cells that supports the germline stem cells, and also the cyst cells that support the germline stem cells. As the germline stem cells divide to form germ cells and further forming the sperm, the cyst cells, the cyst stem cells stop dividing and they start to expand tremendously to enclose these expanding germline cells. So this expansion basically is a very important morphogenesis. If you don't have that, the testes won't work. So what Haiyang has found is that actually lamins plays a specific role in this cyst stem cell and the cyst cells. And if you get rid of it, so this is actually a illustration of using GFP knock-in to mark the wild-type cell and the mutant cell. As you can see, wild-type cell expands its processes to enclose the germline cells, whereas the mutant basically could not expand. The morphogenesis fails, and then cyst cells don't enclose the germline cells, and then spermatogenesis fails. So that's really a very dramatic demonstration of how lamins are involved in morphogenesis. And actually the mechanism Haiyang has illustrated is that if you don't have B-type lamins in these cyst cells, that the nuclear pore complex, nuclear pore protein called the nucleoporin 153 gets aggregated around the nuclear periphery. And then when that happens, then the nucleus are not able to retain the phosphorylated ERK. It turns out you need proper localization of nucleoporin 153 in order to retain phospho-ERK in the nucleus. And phospho-ERK is very important for the cyst cells to undergo its morphogenesis. So that's really the mechanism. That's what we have found to be the mechanism. So that basically shows lamin could use one mechanism, such as ensuring nuclear pore component, proper localization of nuclear pore component, therefore proper localization of phosphorylated ERK in the nucleus to ensure cell morphogenesis, which then help organogenesis. Okay, so that's that highlight, and then I'm gonna go quickly through aging. So basically, the concept really is if lamin is very important for the making basically morphogenesis and organogenesis, then you can imagine upon aging, the major problem is our organs and tissues really cannot maintain the shape. So perhaps lamin could be one of the culprits in aging. And indeed, there is literature on studies of in vitro replicative senescence using fibroblasts, primary fibroblasts. There have been findings showing that if you just take the fibroblasts, grow them, get them into senescence, you can see that there is this phenomenon called senescence-associated secretory phenotype, SASP. meaning that when cells get into senescence state, then it secretes a lot of inflammatory cytokines and chemokines. And accompanying that is lamin-B1 reduction. There's a very clear lamin-B1 reduction by at least half, if it's not more. But this is really an in vitro system, and it's unclear if this is actually happening in vivo. And this is how Haiyang, who's really wonderful fly geneticist, he decided, okay, I'm going to use fly to tackle this, because it's too hard to study this in humans or mouse, because it has too many. It has two immune systems, adaptive and innate immune system, whereas in Drosophila, you only have innate immune system, and you have a fairly simple immune system. The major one, the humoral fat body, is a regulates humoral response of the immune system, and it's just right underneath the cuticle, shown here in blue. And then another local immune system is the gut, Drosophila gut, which is basically bathing in the hemolymph. So Haiyang basically looked through all the tissues of young and old flies. One very striking change he found almost right off the bat is that the fat body cells shown here exhibited very clear lamin-B1 reduction upon aging. But it's really only lamin-B. Lamin-A didn't seem to change. And then if he looked at the gut doesn't seem to change its lamin level. And then to just summarize his work, the finding really he made is it turns out upon aging, the fat body cells really have a very significant loss of lamin-B. Lamin-B, I think it's about at least 50%. And this reduction leads to systemic upregulation of systemic inflammatory factors that get secreted in the hemolymph in the secretory system. And that really led to the local repression of the gut immune response. We all know that in our gut, we have to modulate our microbiome. So you have to have an immune system there to make sure your microbiome won't go out of control. Now, if you have this system, if you have too much inflammatory factors here that represses the local immune system, that would cause the overgrowth of the microbiome, that could cause damage. And then the gut, basically the gut stem cells, try to repair that damage. But the repair is just defective because you are dealing with the old gut. You end up with basically misdifferentiation and increased proliferation and misdifferentiation of the gut stem cells, and that then led to this gut hyperplasia. So this is one way that lamin-B reduction in this case it's post-mitotic cell, whereas in this case it's continuously being replaced. So you don't see lamin-B1 loss. But this loss of lamin-B1 in the fat body could really influence other systems. So now Haiyang took this whole project to his own lab in China, in Zhongshan University, and I have one little project left to show in mammals to have an example of that, and we have an example that's thymic evolution is also in part driven by lamin-B1 reduction in the thymic epithelial cells, and I'm not going to talk about that because I want to really talk about genome organization and transcription, how lamin could play a role in that. So now, why do we really want to look at genome organization and lamin's role? The main reason really is kind of wild. It's kind of a wild idea, but it still remains to be the idea that I'm most excited. That is, if you think about do we really understand morphogenesis? How does morphogenesis occur? We have hypotheses. We have transcriptional cascade, we have signaling cascade, and we have mechanotransduction, right? It's all kind of cascade idea. And I'm a cell biologist. The cascade doesn't make sense to me. I need to look at the morphology. I want to understand the morphological code. And I really felt that this connection between lamin cytoskeleton and lamin with chromatin could be where you could see a morphological code. And for example, if you are exerting force to change the nuclear lamina, then you might actually change chromatin organization. And that then might help you to really understand if you want to become an elongated epithelial cell, you want to have a different kind of 3D chromatin organization by influencing lamin, then you establish that transcriptome, right? So this is basically a dream, that's what I'm saying. So in fact, this dream is not completely unfounded, I would say. We know lamin interacts with lamina-associated chromatin domains. They are called LADs. And in fact, people have found that these LADs regions dissociate, move away from the nuclear lamina region to become internalized. And that internalization shows lineage specificity. So that kind of is hopeful and encouraging. And so the question then is, could the dissociation of LADs shape lineage-specific transcriptome? And another thing I didn't add here because I think it's probably even crazier is, could the interaction between the cytoskeleton and nuclear lamina through the nuclear envelope, the force, kind of change the lamina meshwork, which then lead to the dissociation of LADs? And that could hopefully help you to decide which LADs goes inside, and then determine the tissue building. And we already showed that lamin is important for tissue building. Okay, so, but if you want to do this, it's very complicated. So you have to kind of first understand, if you don't have lamin, what is the change on the genome organization in 3D? So one thing I just want to say is that in the field, in lamin field, because nuclear periphery is considered as a repressive environment. So for years, people say, oh, the nuclear periphery is just, lamin plays a repressive role. And when we removed lamin from ES cells and now from other cells, we really don't see that. So you can look at gene expression change. This is wild type versus lamin triple knockout ES cells. And you can see this is a fold change of gene expression, and you can see the association with lamin B1 using DAM-ID technique. And you see no correlation. You can have upregulated gene or downregulated gene, either with genes that's in the LADs or not in the LAD. So if you don't have lamin in the cell, you don't really specifically affect genes in the LADs. You affect global gene expression. So that's the first thing we learned by doing this clean knockout. But it's really hard to explain that. Is that just basically something you don't understand? You will never understand, it's just random. But we felt maybe it's not totally random. At least it's important to look at what happens to 3D genome organization when you don't have lamin. Well, in ES cells, the change is not dramatic, but it's clear. There are changes. First of all, if you don't have lamin, so this is basically a chromatin, a region of chromosome 10, wild type and laminal cells. And this is a plotting to show that the topologically associated chromatin domain TADs do not change if you look at this too. If you don't have lamin, you don't affect TADs, which actually makes sense because lamin is at the nuclear periphery. It shouldn't affect TADs. But what it does affect is TAD-TAD interactions. So each of this black line basically outline one. So this is one TAD, right? So when you start to compare the inter-TAD interactions, now you can see the change. So basically there's one TAD, there's an increased interaction in TKO cells between this TAD and this TAD on chromosome 10. So you can see this throughout the genome. If you don't have lamin, you are changing the much larger scale chromatin structure. So now, to really understand this better, just looking at TADs is not enough. TADs is just too big of a chromatin structure. You have to really dial it much lower at a higher resolution. Now, to do that, a very talented bioinformatician in my lab he developed a hidden Markov model to try to separate the chromatin domains into different features by using lamin association LADs, as well as histones, H1, H1D, 1C, and H3 also histone modifications, various histone modifications. He's able to separate the chromatin domain into 6 different features. So we kind of laminate highlands, it's histone lamina associated domains. So he used the color from red to purple to kind of just name them. And then the red region, based on the histone modification, is most active, and followed by the next active region is the yellow region, and the orange, green region, highland G, is less active, whereas with this model, he's able to separate the LADs into 2 chromatin features. One is highland B, the other one is highland P. And I just need you to remember this blue and purple are LADs regions, and they are separated by their feature of this blue region actually is having higher enrichment of H3K27 trimethylation, whereas the purple region has a lot more lamin-B association. Okay, so this actually is very important for us to be able to further study the relationship of, how the 3D is changing, because now we can map our highland region to the TADs, right? These are the TADs. And as you can see, the purple LADs, the LADs, purple region of the LADs, oftentimes is really long, which is representing mostly heterochromatin region, of a constitutively heterochromatin region. And oftentimes they could cover more than 1 TAD, whereas the blue region, the blue LADs region are much shorter. Sometimes it could be one TAD long, but sometimes it's less than 1 TAD long. And all the rest of the interior chromatin, the euchromatin region, they are much smaller and often have multiple 1 TAD could have multiple features of this chromatin. Okay, so through this analysis and looking at by in-depth analysis of the Hi-C data, we predicted that when you lose lamin, it looks like the purple LADs region are actually expanding. So this is again chromosome 1, 4, 13, and 14. That's basically, from the global analysis, it predicts that the purple LADs region are expanding. But you really want to show that, so we performed a fish experiment, basically by designing oligo arrays on selected regions like this, and then just do the oligo paint. This is a very cool method which allows you to really look at the volume and surface area of these specific regions you painted. And as you can see, the triple knockout ES cells have significantly expanded purple LADs. All of this, each one of these 4 probes showed the expansion. Whereas the Hi-C analysis, the sequencing analysis, also suggests that the blue LADs region looks like it's kind of fall inward. So again, you can use the oligo paint to show that, and by picking different regions, you can see, indeed, if you measure the nuclear periphery, distance from the nuclear periphery away from your fish probe, you can see that TKO, the blue region, basically shifting inward. So now, this is one finding. So basically, this 3D genomic change seems to be probably starting from the purple region and blue region changing differently. Purple region is expanding, blue region is going inward, whereas in the past, people have been always confused and troubled by the fact when you don't have lamin, you just don't see a clear change on the LADs. Here, our data really suggests, well, the reason you don't see that is because you are looking at LADs as a totality. You are not separating them because they are changing differently. And I will give you a model toward the end. So the other thing we tried to do, we are able to do, because we are able to really look at the chromatin regions with its features, is to really look at enhancer activities, the active enhancer, H3K27 acetyl peaks that represents active. It's correlated with transcriptional activity. So you can look at the interaction changes between these chromatin regions and then see how it's correlating with the change of enhancers. And basically, the take-home message for this part of the figure, so here basically repeats showing you there's no correlation between lamin association and gene expression activity. But we do see some correlation between active enhancer change. For the enhancers, that's upregulated. And these are chromatin regions that show a dissociation from this most repressive LADS region, the purple LADS region. Whereas for enhancers that's downregulated, we show an increased interaction with this other inactive region, the blue LADS region. So this really shows the only way you can understand the gene transcriptional change when you don't have lamin is to understand the 3D genome interaction change. And indeed, I can also show you a specific example here. So basically, here is to look at two specific loci. One is Ryan locus. It's downregulated in TKO ES cells. Another one is NOD1 locus, which is upregulated in ES cells. So again, so this is actually a 4C analysis of across this region using a probe, by looking at the interaction with this Ryan locus. And this is basically the highlands that I have been talking to you about. And then this is basically a high C analysis. So what I want you to look at is, so this is a Ryan locus, and that's downregulated. What you can see is when you don't have lamin, this Ryan locus exhibit an increased interaction with this inactive chromatin region. That's blue. This is a blue LADS that's inactive. Whereas it has a decreased interaction with this highland red region, which is the active euchromatin region. So that could explain why this is downregulated. On the other hand, if you look at this upregulated NOD1 locus, you can see that probably one thing that contributes to its upregulation is because it's picking up it's basically starting to dissociate. So you go up from here, look at this decreased interaction. Blue is decreased interaction with this purple LADS inactive region, and second one and third one. So this really is correlated with basically when you have a LADS change and you have TAD interaction change that makes the chromatin to find itself in different neighborhood, different activity region. That can explain global effect on genome on genome-wide transcriptional change. Okay, so I'm going to give you a model, and then I will spend a few minutes to just kind of entertain a couple of ideas. Well, one idea essentially. So basically, the take-home message is the traditional understanding of nuclear lamina is always, nuclear lamina is a tether for the lamina-associated chromatin. But what we found is if you remove this meshwork, you don't have wholesale inward folding of the LADS region. Instead, this purple region expands, and we believe this expansion basically makes no space anymore for this blue LADS region. Blue LADS region, we draw it in a more condensed manner. It's because they are more enriched for K27 trimethylation, which has a role in condensing chromatin. So we think this region falls inward. We do have data to show that. So when this region falls inward, then it basically changes the chromatin interactions. And then if you push this gene, if you push this gene that's expressed into a way more kind of toward the repressed purple region, it's going to be repressed. And then if you push this inactive region away from this purple region or push it closer to this active chromatin, then it becomes expressed. So that's why the impact is really genome-wide. Now, why is this important why is it important? That's the first question. Why am I excited about this? Well, the reason that's exciting really is, again, you come back. So we said, basically, if you remove all the meshwork, you have this dramatic change of chromatin organization. Now you can imagine, we know, in fact, from other studies I didn't have time to talk about is, when we remove all lamins, you can see the nuclear pores all aggregate toward the centrosome because the dining is pulling all the pores towards the centrosome. So lamins really have a role in kind of making sure that the underlying, whatever force that cytoskeleton is exerting in the nucleus is modulated. So now you can think perhaps as cells are exerting force to kind of build, moving and exerting force along the nuclear surface, you could imagine that force could help to change the mesh size of the nuclear lamina. When that mesh size change, it's almost like you are just locally removing some lamin mesh. That could locally open up this purple region. That could then allowing the neighboring blue LADs to fall inward. And then you know that cell morphogenesis often is transient, and different trajectorial differentiation could have impact on different regions of the nucleus surface. So that could actually one day perhaps help us to understand how this kind of morphogenesis would have perhaps direct impact in how you establish 3D genome organization, therefore a unique transcriptome that's lineage specific. So I know, probably not in my lifetime we're going to understand this, but at least kind of this journey for over 10 years of looking at lamin, so far so good. Maybe at some point it's going to fall completely wrong, but so far it looks like lamin regulates morphogenesis, and lamin really connects cytoskeleton to chromatin. And then if you don't have lamins, you could have this significant inward falling of chromatin that will lead to the global genome organization change, transcriptome change. So perhaps this could be a node that will allow us to understand the morphological implications of how you organize different transcriptome. That's it. So I'm going to quickly acknowledge people in my lab. I think I'm doing okay with my time. So the cell division study is really a long-term coming. I start my lab to try to understand cell division. We actually did some gamma-TERC work, microtubule-nucleation work, but we decided not to go do more because there are a lot of good people doing that very quickly. So we mostly focused on spindle matrix because not that many people are doing it, and that's where it's better to work on this thing. I believe it's very important. So, and many of these people contributed. I think no one is currently in my lab now. I'm actually trying to recruit a mitosis person to work in my lab right now. So, and then the genome organization work and also aging are mostly done by this group of people. Jabiel did a lot of FISH for the work I just told you. Xiaobing was very important in helping us to analyze and model the chromatin, and Haiyang did the aging work and all the Drosophila work, and Yongzhou did a lot of work on the initial establishment of the system. And I've had a number of collaborators, and some of them I'm still collaborating. And here are my funding, and I finished my Allison funding, but that was actually very important for us to do some work in aging and lamin. Okay, thank you very much.
We have time for some questions. If you could please go to the microphones. Yes, here.
Thank you. Very interesting talk, very intellectually exciting.
Thank you.
Although the tools of science has been removed from me, but the love of it has remained intact. My question is, you mentioned the three stages of life, from embryo and the spindle shape all the way through the senescence, and then you touched a little bit on the diseases. My question is whether this protein structure of the tubule or laminin changes conformationally as far as perhaps the proline that you mentioned, it's going to conform in some way. And or is it later dependent on the other protein structure such as, branch amino acids like alanine? We saw something years ago at the University of Pennsylvania, but that was related to myotube to myoblast formation.
Okay, so are you specifically asking about the phase separation of the bug Z protein and what amino acid does proline play a role?
In a way, are you changing the structure of the protein from embryonic and the mitosis at that stage, which is a very formal growth, then in the disease stage, which is a chaotic?
Right. So yeah, I guess one good example to use would be lamin. As during development, you definitely need lamin to help you to maintain the structure and to support morphogenesis. And then as you get older, the protein just starts to decay. We know, by studying the fat body in Drosophila and thymus, we know some cells basically when they go into senescence or when they stop dividing for a long time, lamin B1 level just becomes reduced. And that reduction in the fat body case leads to the de-repression of the inflammatory chemokines and cytokines. But basically this is all downstream consequence. The question is, how does lamin B get reduced? We actually still don't know. We did a lot of work. We still don't really have a very good clue about why lamin B1 become reduced upon aging. It could be regulated by ubiquitination. It could be. We don't think it's necessarily regulated by transcription because we don't see transcription.
Senescence.
Huh?
Senescence, maybe.
Senescence. Yes, senescence is the biggest trigger, but still, it's not a direct. It cannot explain how lamin gets reduced. Right.
Thank you.
Thank you.
Yamini.
It was a really great talk, Zhang. So I have two questions. One is about the bug Z protein. So you had this awesome in vitro assay where you showed it can very nicely phase separate when you raise the temperature, right? So I'm curious, what if you take like interphase extracts versus mitotic extracts? Does it care what the components are in terms of salt or calcium or whatever that's present in the interphase versus the mitotic extract that would, so that's a signal for it to start phase separating in vivo, to get some insights into that?
Right. So, I think that's in vitro is just an artifact where we just, because we only have one protein. It's like tubulin, right? Tubulin is a temperature. Tubulin polymerization in vitro is temperature dependent, and in vivo it is too. If you cool down the cells, microtubule just depolymerize. Right. You can think about bug Z phase separation as just tubulin, like tubulin. It just because. In the in vitro assay, in that sense, you just really have one protein. You don't have regulators. But in vivo, even in vivo for bug Z, it's also is similar to tubulin. We've done this experiment by taking tissue culture cells and then warm it up. Well, we cool it down on ice. You can see bug Z association with microtubule is decreased. So I know what you're asking is, how can you explain this temperature dependence?
Actually, no, that wasn't my question. My question was simply, is there some chemical property within bug Z's folding domain which is sensitive to some salt or some cation that's released during mitosis, or because the nuclear membrane breaks down, and that isn't present during interphase, that catalyzes phase separation specifically at the right time of the cell cycle?
Oh, yeah. So, actually, Johannes here, when she was in my lab, this is a nuclear protein in interphase. In interphase, it does have a role in regulating splicing. Okay. So we suspect the interphase is already undergoing phase separation.
Okay, so that's...
And in mitosis, it just undergoes phase separation with different partners.
The second question is just very quick. Really beautiful last story on the TAD-TAD interactions. Right. So one thing that's not clear, though, is that are you saying that these TAD-TAD interactions are self-organizing, right? Because that's essentially would be the implication of what you're saying, because when you lose the lamin-B, right, the TAD-TAD interactions change, and then you get this odd artifactual interaction between domains that aren't supposed to. So it's almost like PEV. But is this thing heritable? Is this change in the inter-TAD interactions heritable over the cell cycle? So if you compare G1, one G1 to the next G1, do you see the same thing?
Yeah, that would be really interesting to do. I mean, but we haven't, this is basically a whole population. We have not done this cell cycle stage
thank
yet.
you.
No, that would be a very good thing to do.
Well, you covered a lot of different areas of molecular association, dissociation, phase separation, but ultimately you need the force to separate these chromosomes. So do you have simultaneous increase of the ATP to separate in terms of the biomechanical force? You mentioned, I guess, in one sentence somewhere there.
Right.
So do we know enough? This molecular association generates enough energy. What is the source? Do you have a higher level of production of ATP by mitochondria during this separation?
Yeah, that's actually a very good question. I have my collaborator in China. There was one study. That's exactly what they're trying to do. They believe when cells go into mitosis, there is an increase in a certain type of ATP production that's still mediated by mitochondria, by one of the transporters. I don't remember the detail. I mean, I can't even tell you exactly what it is. But yes, there are indications as cells go into mitosis, there's increased production of ATP, and that's helping to supply the ATP for many motors, right? To get cells to divide, there are a lot of motors that need to be very active. Moving chromosome takes thymine and many kinesins to work, hydrolyze ATP to work to pull the chromatin apart. And then you have cytokinesis that needs additional motors to have a pinch into two cells. Yes, I think definitely, I think that idea is on the right track. You need to produce more ATP.
As I was looking at the video of the separation of the two cells, they did not look symmetric. I saw one of them is smaller and the other is larger. And so is it true what I see there?
Well, so actually, I'm sorry, I probably didn't explain well enough. So you are looking at a pseudo-spindle formation where there's no cell, there's no DNA. All we have is putting a microtubule organizing center as the Aurora-A beads, and Aurora-A beads put in there, it forms a pseudo-centrosome. And then they nucleate microtubules, make a pseudo-spindle without DNA. So you are not looking at cells. You are looking at the yellow ball are beads. They are the same size. And on one hand, I had this enlarged spindle, one spindle that's elongating. On the other, on the your right hand is a smaller spindle because you are seeing many more. Yes, I'm sorry, I confused.
All right, thank you. Good luck.
Thank you.
All right. So let's thank Dr. Zhang for a stimulating lecture.